Okay, everyone, so it's a couple minutes past 10 Eastern, so we'll go ahead and get started. Good morning to everyone, or good afternoon, depending on where you are. Thank you for joining us for this webinar. It is day one of the two-day webinar, so please come back tomorrow, and we'll have more speakers to talk about the Heart MPS systems. So I'm just going to do a brief introduction to the 3Rs Collaborative and this webinar series. First, I'll start off with a disclaimer that I'll just read straight from the slide. This webinar and workshop series is made up of companies who may be competitors or customers of one another. Accordingly, nothing discussed during these webinars and workshops are intended to result in an agreement on price, exclude suppliers from any market, or otherwise restrain competition. Those participating in this webinar and workshop series are instructed to avoid discussion of competitively sensitive subjects, including costs, prices, sales, product marketing, and other confidential information. In addition, neither the 3RC nor the IQ will use contact information received during this collaboration for marketing purposes or engage in marketing or sales conduct during collaboration activities. All right. So I'm Eliza Stevens. I'm the program manager here at the 3Rs Collaborative, and we have an MPS system initiative which seeks to increase adoption and regulatory use of MPS. We have over 49 member institutions and over 100 members. Here's the logos of all of our member companies. Our initiative aims to provide thought leadership and expert consensus from developers. We facilitate appropriate discussion, collaboration, and sharing between commercial developers, create direct engagement between developers and regulatory agencies, develop external partnerships and collaborations to provide perspectives from developers throughout the field, and create resources to facilitate increased engagement with MPS developers. We have a regulatory work stream which publishes papers, hosts meetings, and does presentations related to MPS qualification. A technology expo, we create, maintain, and promote the tech hub that we have, and then the end user education. We create educational resource hubs, we host these webinars, and engage with end users in other ways. So for our end user engagement subgroup, last year we had a pretty productive year where we did 4 workshops. We also did quarterly submissions to All-Text Corners and had posters at a number of conferences. This year we'll continue with the quarterly workshops and webinars and posters, as well as our quarterly submissions to All-Text. And we're also working on a consultant panel webinar. We have a presentations page on our website. If you go and look for that, you can find all of our past webinars that we've done in a convenient little playlist. So it's under the events tab. Our regulatory group last year had a pretty productive year as well. They continued forward progress on a cross-platform DILI project, as well as starting a benchmarking project. This year, the iSTAND LOI was accepted for that DILI project, so congratulations to all involved in that. The process paper is nearly submitted, and they're working on a results paper, as well as the qualification plan. They did launch the benchmarking survey on the use of MPS, which I believe closes tomorrow, so if you haven't done that yet, please go fill out that survey. Megan had an invited talk at this Reagan Udall Foundation workshop, and there will be more workshops and posters and talks at various conferences this year. So we have the MPS Tech Hub on our website, which provides we have 32 MPS companies by tissues or diseases or species. We also have a publication hub where we have over 350 publications with improved filters, so you can search through all those publications in an easy way. For the upcoming webinars tomorrow, like I said, we have day 2 of the Heart MPS webinars, and then upcoming webinars are in May, September, and December. Please submit all your questions via the Q&A feature on the Teams. We have a Q&A panel at the very end of this webinar, and if there's time after each speaker, we'll answer a couple questions. All right, so you can visit our website to learn more, and you can always email me if you're interested in joining our MPS initiative.
The goal of this group is to provide a venue for appropriate cross-pharma collaboration and data sharing to facilitate the industry implementation and qualification of MPS models. Yeah, so as you can see, there's 4 main buckets to our group. There are, do you guys see this slide?
Yes.
Okay, great. Okay, so there are 28 member companies with over 150 active participants within the IQ MPS. And we have a diverse representation of drug safety, 3Rs, ADME, as well as PK/PD. This year's executive committee include Rhiannon David from AstraZeneca, who is the chair, Kim Coleman from Genentech, who is the current chair, Phil Hewitt from Merck, who is the vice chair, and James Gossett from Pfizer, who is the vice chair elect. The executive community within the IQ MPS structure provides leadership and guidance, and it implements strategic plans recommended by the steering committee. This is comprised of 2 members from each member company, and they are responsible for primary decision making for the IQ MPS. There are 4 major work streams, which I will go into detail in the upcoming slides, and then a subset of teams beneath that as well. The first stream is the regulatory engagement work stream, and the aim is to create focused engagement between industry and regulatory agencies on the current status and evolving field of MPS in an industry setting. Some of the strategic objectives include the common understanding of these performance standards, building regulatory confidence in these models, and encourage the development of animal MPS alongside human MPS qualification. And we are engaged with in North America, Europe, UK, as well as in Asia. The second work stream is the organotypic manuscripts, where we aim to serve as a thought leader for both MPS developers and stakeholder organizations in the industry for the implementation and qualification of these models. As you can see, we have published a variety of different manuscripts providing an overview of the currently available models, cell types, model features, etc., for a variety of different target tissues and organs, ranging from the lungs, kidney, to cardiovascular, as we will hear more about today. This is the pilot project workstream, where we will provide an avenue for appropriate cross-pharma collaborations. And the goal of this is to deliver on the ability to collaborate and accelerate qualification of MPS. So there are 2 groups, two categories. One is the request for information, and one for request for proposal, where respondents are invited to submit proposals based on the study design provided by the IQ MPS. And we engage with external developers such as Altus and HSI. The SPAC, or Strategic Partnership Communications Workstream, is where I sit, and our goal here is to develop external partnerships, collaborations, and to support MPS adoption amongst the next generation of scientists. And we have external engagements with 3Rs, 3RC, as well as NCATs here. Yeah, so those are my slides. If you have any questions as to what the IQ MPS does, please feel free to reach out directly via email. You can also check out our website, you can see on the right, or our LinkedIn page in the middle. Thank you.
Thanks, Richard.
Thanks a lot for inviting me for speaking today. This 3D engineered heart tissue contractility platform that can be different applications. So as we know, to model heart is quite complex. You need to keep a 3D structure, but also you want to measure the functionality of the heart. And the contractility is one of the main functions, and the pace, the beating of the heart. And this can be used as a readout, for example, for looking at cardiotoxicity, but also to study drug efficacy. And we know that there is a lot of challenges to translate actually the safety and efficacy of drugs towards humans, and there has been a lot of development ongoing to use human relevant models. Also, really integrating functionality and 3D structure, I think this is where we come in. I would like to introduce our platform where we can model 3D engineered heart, but also continuously monitor function of the heart and also be able to pace it, so be able to stimulate the 3D engineered heart. So we are able to make those 3D engineered bundles in 34-valve plate format. Our system is integrated with stimulation plates, so you can also continuously pace your tissues and also place it in incubator and perform long-term experiments. So you can, for example, measure the force development over time. You can also pace your tissue and look at force frequency response. And after the long-term culturing and functional data, the tissues can be collected for further molecular analysis. So why our system is unique, and we think that can bring a lot of interesting results and impact in the cardiac research. So first of all, we can use different type of cells, cardiomyocytes, iPS-derived or immortalized. We can create those 3D tissues that grow between two cantilevers. And one of the unique features of our technology is that one of our cantilevers is flexible, and it's made from stainless steel, so it already applies mechanical load of your tissue. And then when the tissue starts contracting, we are able to measure it with our with the lights. We have a sensor, and then it creates very unique and very high sensitive way of measuring the contractility. So we'll provide you a lot of sensitive data and kinetics of the force. As well as our lead is composed of multiple components, which are integration of stimulation plates, so you can do pacing inside the incubator and do the measurement of the force. There is also 24-valve plate format, so it gives you some scalability in terms of measurements and culturing. Today I want to show you some data and highlight the interesting results that we received with our platform from our users, but also within the partnership that we created. So first of all, we can really look at mechanistic relevance of the heart. For example, if you look at molecular, you download certain gene, and then you can look at the functionality and how that results. Furthermore, we can combine pacing with functional controls. You can pace your tissues, but also look at the output, how you measure the force. And latest, we want to design an assay that is standardized, so you are able to use commercialized cells with our system to look at drug safety and efficacy. So this is an example of the recent publication that we got with our core system, where King's College and John Carlo Forte created knockout line, YAP knockout, which is protein that is involved in embryonic development. And when you see that if there is lack of this protein, you see that force development there is no increase in force over time, while in the control you do see a nice increase of the force when a tissue matures. But if you, for example, if you look at the slide here, if you reintroduce YAP, there is also restoration of the force. So this way you can link a function of certain protein or gene to the actual function in the heart. And then if you take out the biopsies and look at the structures, you also see, you can appreciate that in those knockout cells, you also see that the sarcomeres that are responsible for are not properly formed. And if they quantified it, they also see that the sarcomeres are much shorter than in control lines. Also, another collaborator just recently published paper where we looked at triple cultures and wanted to combine pacing and measurement of the force. So what you can see here, the 3 cell types were derived from IPS and cultured together. So we have here endothelial cardiomyocytes, cardiac cardiomyocytes, endothelial cells, cardiomyocytes, and cardiac fibroblasts. They're all mixed together in collagen microgel, hydrogel, and then the tissue is formed. And here you see beautifully the compaction over 48 hours, that they compact around the pillars. And then the tissues were continue. Here you can also appreciate the nice distribution. So you can look at the troponin. This is the bank of cardiomyocytes, cardiac fibroblasts, fibramented, and endothelial cells. So they're all nicely distributed within the tissues. And when the tissues were here, you can see what we have done. So here within a quarter system, you can measure spontaneous contractions over time. So starting at day 4, and here are the normalized data. So you see that the force increasing at certain way, stabilizing at day 16. And we can measure spontaneous contraction, but we also can do a pacing. So we can pace the tissue at 1 Hertz and also look at the increase in force over time. Also here you can look at the blue bars represent continuous pacing. So the tissues were paced at 1 Hertz for 3 weeks. And then the tissues that were not paced in gray. But because we paced at 1 Hertz, physiologically relevant, you don't have a difference in force development between stimulated versus non-stimulated tissues. What is also very important mark of maturity is actually the force frequency response. So if you start seeing a positive force frequency response, so if you increase the frequency and then you start seeing that force of contraction is also increasing, that it's a sign of maturation. And this is what we see actually on this slide. So at day 4, you do not see the force frequency response, while at day 16 and 18, there is a clear increase at higher frequencies. Data we have shown that we are able to culture 3D more mature tissues, and we can also pace them continuously, and also look at different type of measurements like force of contraction, but also look at force frequency response. And last but not least, we conducted a collaboration work together with commercial company that develops cardiomyocytes, Axel Biosciences, in collaboration with King's College as well, with protocol in which we co-cultured cardiac fibroblasts and iPS-derived cardiomyocytes derived from Axel, and we were able to look at longevity and maturation of the tissues. And what you can see here, this is 24 tissues cultured for over 45 days, and you see a nice increase in force over time, and that they are able to be kept at over 45 days in culture. And you also see an increase in force, and also spontaneous beating frequency goes up, meaning that the tissue matures over time, becomes stronger, and also beats faster. After that, we also looked whether these tissues respond to drugs, and we performed isoprenaline challenge, in which we increased concentration of isoprenaline, and also at the same time we pace the tissue so they synchronize and pace at the same frequency. And then what we see that with increase of isoprenaline, we also see an increase in force of contraction, which is an expected response to this certain drug. So this is with this I'd like to summarize to say that with our system you're able to look at mechanistic studies. For example, if you're interested in certain knockout of certain protein and see how that affects the functionality of your tissues. If you're interested in looking into maturation of your tissue and see how the pacing effects or exercise of my heart would affect the function. Or we also provide protocol together with the cells and instruments, so you are able to enter the 3D space, but also be able to look into more standardized way of doing 3D engineered heart tissues and measuring effect on different drugs. So with this, I'd like to conclude and thank you for your attention. And if you have any questions, please let me know. And if you are interested, please contact me. Here's my address. Thank you.
Thanks so much, Svetlana. We do have one question for you. How long does the primary cardiomyocyte constructed 3D bundles last? 30 days or longer?
Yeah, so it's we showed with AXL cells, we used 70% iPS-derived cardiomyocytes and 30% cardiac fibroblasts. Those cells last 45 days. But we could have done even longer experiments, but we stopped at that time.
Great. Thank you so much.
Thank you. Hi, everyone. It's a pleasure to speak to you today. My name is Lizzie, and I'm the Business Development Manager and U.S. Technical Lead for AIM Biotech. And today I'm going to share a couple of examples of how AIM's Microphysiological Systems have been able to support the development of vascularized human cardiac models. And so just in case you're not familiar with us, we are an MPS company that was founded out of Roger Kam's lab at MIT, with our commercial operations headquartered out of Singapore and led by Quan Chi Moon, who co-founded the company with the support of the MIT Singapore Alliance for Research and Technology, also known as the Smart Innovation Center. So over the past 10 years, we've done a lot of internal R&D, but we've also had a lot of collaborations with Roger's lab at MIT and partnerships with biopharma companies and the that we have and a lot of different models, some of which you can see here. And really how this has been done is just by enabling researchers to be able to easily recreate some of these more advanced 3D biological processes and tissues, including, important to today's talk, the development of perfusible vascularization of different tissues. And you can do this in things like the blood-brain barrier, but you can also vascularize different types of organoids. And today, of course, we'll talk about how that's been applied for cardiac organoids. This biology is all created by using the sort of simple 3-channel microfluidic design, which can be scaled from 3 experiments on a glass slide up to 40 experiments within a plate. And then within our newest platform, which we call organics, this is allowing you to culture much larger tissue samples up to 2 millimeter diameter through this open port on the top. And we'll talk about how that's been applied in cardiac modeling. And so just to, before diving into the data, I just want to talk about what is the need for vascularization within a cardiac model. What we would say is that the vascularization process is important because it is very essential for proper functioning of organs and tissues. It governs the viability of each tissue through the transport of oxygen and nutrients and the removal of waste byproducts. And within the heart, crosstalk between endothelial cells and cardiomyocytes plays a role in cardiovascular diseases, making it a necessary component to better reflect the in vivo tissue architecture in response to therapeutic interventions. And then finally, vascularization of cardiac microtissues or organoids in vitro can help stabilize the tissue and extend culture duration. And so microfluidic 3D systems such as AIMS have been shown to support the generation of perfusible microvascular networks, which again, we can do within Identics and Organics, and we'll talk about a few examples within those platforms. To understand how the vascularization process is achieved, just wanted to quickly provide an overview first of Identics, which again is available in a few different formats. So you can have 3, 9, or 40 individual tissue sites being cultured simultaneously. And the way that each tissue is cultured is by having the 3-channel design. Within the center channel, you have the hydrogel, which can contain cell types. And then you have 2 flanking media channels, which allow for media perfusion, addition of any sort of exogenous chemokine or cytokine, as well as if you had a drug specifically that you wanted to add, you could add that through the media channels, and that can interact with the tissues being cultured within the center channel. The perfusion is actually driven mainly by gravity, but if you wanted to have continuous flow or very specific shear stress on your tissues, you can add lures to Identix 3 and 9, which can then be connected to syringe barrels for extended gravity-driven flow or through tubing to off-chip external perfusion systems. And all of the platforms are SBS compliant with 384 well footprints. So that means that you're able to have automated liquid handling to set up the experiments, and then you can do all your imaging in situ and automate that as well. This 3-channel design does allow you to have concentration gradients and pressure gradients to be established, which is important to the process of vascularization. And then finally, I just wanted to mention that the bottom of all of our cultureware is gas permeable, so that you don't have to place these plates on any sort of plate rocker or have the external perfusion. So vascularization can take place without that, and you can just keep these in your incubator, just sort of in the standard way that you would maintain culture with any sort of well plate. Okay, so with that platform in mind, I wanted to walk through how research teams out of Leiden University Medical Center, led by Drs. Christine Mummery and Valeria Olava, use this Identix platform to develop a fully vascularized and perfusible cardiac microtissue on chip. In this study, the team co-cultured human-induced pluripotent stem cell-derived pre-vascularized cardiac microtissues with exogenous vascular cells within a fibrin hydrogel, which then allowed them to study how endothelial cells communicate with cardiomyocytes through paracrine signaling, and how this can then have downstream implications for response to drugs and inflammatory stimuli. So using a previously established protocol, the team first generated the pre-vascularized cardiac microtissues, which, as I mentioned, then were mixed with additional human iPSC ECs and supporting stromal cells within a fibrin hydrogel in the center channel of Identix. And as you can see here, over the course of about 5 days, they were able to achieve robust vascularization, and you can see that vascularization actually happening within the first 24 hours. You start to get interconnected microvascular networks formed around day 3, and the networks become stabilized around day 5. And if you zoom into the cardiac microtissue, you can see how with contraction, you get a constriction of the blood vessel. And then additionally, you can perfuse these with fluorescently tagged beads, so these white dots that are flowing. And so that with every heartbeat, you can see how those microvessels are constricting and the beads are flowing through those blood vessels. To compare the dynamics and model the endothelial cell cardiomyocyte crosstalk, the team compared cardiac microtissues with and without the extravascular cells in the fibrin matrix. So those would be the two conditions shown here. First, they observed how there is slowed beating, or in other words, longer culture duration and relaxation time for the vascularized microtissues. And the team quantified that shown here. Then they added a non-selective inhibitor of nitric oxide, which is referred to as L-name. And after 6 hours of incubation, it was able to reverse that slowed beating and decrease contraction duration and relaxation, but only within the microtissues that had vascularization. So those that did not have vascularization of that inhibitor. And this finding really underscores the importance of having vascularization because this response is only present when you actually have the endothelial cells within the system. And that demonstrates a more faithful representation of how a cardiac tissue is really in vivo. In another set of experiments, the team modeled the response to the addition of inflammatory cytokines, so IL-1 beta. And similarly, only the vascularized microtissues showed a response to the inflammation through upregulation in the secretion of IL-6 and cytokine chemotactin protein 1, again suggesting that the vascular units were the only part of the model that were affected and responding to the pro-inflammatory cytokine stimulation, which are known contributors to EC dysfunction and heart failure. Additionally, the vascularized microtissues show decreases in contraction duration, relaxation time, and peak-to-peak time. But this, again, was only present for those that did have the vascularization present in the cardiac microtissue. And this finding actually was somewhat unexpected because IL-1 beta and IL-6 have been shown to increase nitric oxide levels through upregulating the expression of inducible nitric oxide synthase in isolated cardiomyocytes in previous work. But on the other hand, IL-1 beta and IL-6 decrease endothelial nitric oxide synthase expression and activity in ECs. And so the team didn't go further into looking at the expression of iNOS and eNOS upon IL-1 beta treatment. But the fact that IL-1 beta inhibitory effect was only observed in the vascularized cardiac tissues indicates that there's an important importance in incorporating the vascular compartment in mediating the response of the cardiomyocytes to these stimulations. And so taken together, this platform opens up the future opportunities to model selective nutrient and drug delivery through the endothelial cell barrier to target the cardiac tissue, therefore improving the disease modeling and the ability to effectively predict drug efficacy. And so while the IdenTix platform was very sufficient for culturing these smaller microtissues, some researchers generate larger cardiac organoids that would not be able to fit physically within the IdenTix platform because the height of the channels is only 250 microns. And so as cardiac tissues become larger, that need for vascularization becomes even more important. And so just before showing kind of how that can be achieved, I want to just highlight that the reason you would want to have vascularization of larger cardiac organoids is because you would like to ideally avoid any sort of necrosis that can occur within the center of organoids when the oxygen tension starts. The vascularization also can support larger organoid growth for extended culture durations, as mentioned, and so that can improve disease modeling and the drug discovery processes. And then additionally, the viability of implantable organoids is improved when vascularization is present for regenerative types of approaches. And so to do this in vitro, users can use the organics platform, which, as I mentioned, has this opening on the top. So that opening is 3 millimeters in diameter. And so tissues that are about 2 millimeters in diameter can still be embedded within a 3D hydrogel in that center compartment. Similar to IdenTix, there are, again, 2 flanking media channels on either side of that center hydrogel compartment, where you can have the endothelial cells and supporting fibroblasts in order to vascularize different organoids. And again, this platform, which features 24 independent tissue sites, is also SBS compliant with a 384 well footprint. The meniscus pinning actually between these compartments is what allows you to have that 3D hydrogel adjacent to the fluid phase media channels. And then finally, because there's gas permeable laminates along the bottom of this plate as well, you don't have to have any sort of plate rocking or off chip perfusion in order to achieve the vascularization. And one really unique piece of this platform is that you can actually extract the tissue at endpoint intact and embed it in something like paraffin if you wanted to do further downstream histological sectioning and analysis. And so briefly, with a early collaboration with Oscar's team out at Stanford, we were able to demonstrate some early success in supporting vascularization of his larger cardiac organoids. His team recently published this paper on how they are able to generate these organoids from human pluripotent stem cells through spatially patterned cells that are then able to characterize the gastruloid, progenitor, and cardiovascular cell type formations. And these organoids can, again, form their own internal luminized blood vessels, but it's a kind of a challenge to actually continuously perfuse those lumens and maintain stable vascular culture without having additional vasculature around each of those organoids. And so therefore they look to use organics to add those additional vascular cells around each organoid and have that anastomose with the internal lumens present. And in addition, in this early collaboration, they are also testing out AIM's new vascularization kit called the VasQ kit, which basically is sort of like a DIY kit that allows you to do the vascularization process in a more reproducible manner because we provide the cells, the ECM and media supplements, and as well as the platform in order to achieve vascularization of your tissue of interest. And shown here, this was with Thermo Fisher and their patient-derived tumor organoids being vascularized in organics with the VasQ kit. And so just as a control and to confirm that the team was able to actually vascularize tissue, they set up an experiment without any cardiac organoids, and they were able to show that these blood vessels were both perfusible and were able to express the important cell-to-cell endothelial cell marker CD31. And with cardiac organoids, they were able to show that those organoids are able to be functional still and actually beat in both the unvascularized cardiac organoid setting as well as the pre-vascularized cardiac organoid setting. And then finally, the team was able to use immunofluorescence staining to confirm that healthy vasculature was able to positively express CD31 and that these vessels can actually integrate in and around the organoids. And these vessels are also perfusible, and this can be shown through the perfusion of the fluorescently tagged dextran into the system. So although these results were quite preliminary, the proof of concept was able to be established through their first experiment where they could culture these organoids in organics using the vascularization kit, the VasQ kit, to form the vasculature. And this will then help support the team in creating more mature cardiac tissues and allow them to have more realistic delivery of therapeutics. And so in summary, I just wanted to share how the researchers, different research teams can use either identics or organics to achieve vascularization of their cardiac tissues. And reproducibility and standardization can be more supported through using something like the VasQ kit to add that additional vasculature around organoids. And so I'll stop there and say thank you for your time, and please feel free to reach out to us, follow us on LinkedIn, and I would be happy to answer any questions.
All right, thanks so much, Lizzie. We don't have any time for questions right now, but feel free to put any in the Q&A box for her later.
So those of you who are familiar with Hesperos, it was founded by Mike Schuler and I. Mike's considered the founder of the body-on-chip field. We've licensed a bunch of patents from where I'm a professor at University of Central Florida and Cornell, where he's a professor. We had won Lush Prize specifically for animal testing. We basically work with folks all over the world. We got a 14,000 square foot facility here in Orlando, Florida. We got about 50 people at the company. We are CRO. We don't sell our systems. Mike and I decided early on they were too complex to be able to do that. But we have a full service CRO where we can do everything from planning through all analytical capabilities. But also, we also are the first microphysiological system using efficacy data to repurpose a drug for Sanofi into an IND for a rare disease that has been successful, and it's now in a 2-arm phase 3. So what do we do differently than most folks? We've been working on the idea of clinically relevant functional readouts for about 30 years. So what does that mean? So you go to a doctor's office, they don't immediately stick a needle in your arm and be able to put fluids, look at biomarkers, right? Like how you're walking, how you're talking, are you making sense? They listen to your heart. They check your reflexes. They might ask you about elimination. What they're really checking on is the muscles contracting okay? Is the electroactivity of your neurons and cardiac cells functioning properly? Is your neuromuscular junction working well? Is the barrier integrity of things like your GI tract and kidney functioning? So what I've been doing is for that time is take silicon devices, use surface chemistry to integrate the cells with those devices. So say when cardiac cells contract, they bend the silicon, which then deflects a laser, and I can get force. I can pattern cardiomyocytes over top of microelectrodes and be able to get conduction velocity and QT interval. Again, same way and get skeletal muscle force. I'm not going to talk about them today, but we have a wide variety of different peripheral nervous system and central nervous system assays. And then we can still look at biomarkers for things like liver, but we don't have to for all the different organ systems. This allows us to non-invasively look for acute, but more important, chronic monitor these systems. We published a 4-organ system out for 28 days. We can go further than that with many of the systems as well. But we're also trying to do this without cell death. We really want to have this clinically relevant aspect where what we're measuring in our systems correlates to what a clinician would be measuring from a patient. We can then culture out the cells on their own individual media, on their own chips, then time it to assemble them all at the same time, be able to put a gasket over top of it to be able to look at the fluid flow, everything is fully immersed in the serum-free medium. And again, I had banned the use of serum in my lab about 25 years ago, so everything we're doing is serum-free. We can also then put a clear top on it, do all the advanced imaging. So say if you've been using a cardiac organoid and you want to see how that compares to our more functional models, we can do that fairly straightforwardly in our system. Just quickly, we repurposed about 9 drugs I can talk about into clinical trials using our systems for sarcoptic tooth disease with Vanda, ALS with Apellis, ALS with Argenx, myasthenia gravis with Dianthus, which has now been successful and they're now raising money for phase 3, and also MMN and CIDP with Dianthus, and then CIDP and MMN with Sanofi, and those are the ones I can talk about. So we have a wide variety of different CNS systems and PNS systems where we can also look at efficacy. Now, Mike had basically taken the equations for predicting phase 1 dosing a long time ago, and then modified those to be able to create the first multi-organ system. This was published in 2004. This is one of our more advanced systems looking at opioid overdose and recovery in naloxone, at the same time looking at off-target tox. That's recently just published. But also, Mike also introduced in 2010 this idea of a pumpless platform using gravity to pump it. This gets rid of all the tubing, all the external pumps, but also more importantly, makes it a low volume system. So I can look at the effect of parent and metabolite in the same system if a liver is present. So as I mentioned before, we can use surface chemistry to be able to pattern cardiomyocytes, be able to get rhythm generation, conduction velocity, and AP length at QT interval. And on a separate chip, we can get force independently. So we can get relative force, but also absolute force off of these systems to be able to predict cardiac output. This was first published in Biomaterials in 2015. We have shown in our serum-free medias that Fujifilm cardiomyocytes can get maturation over a period of about 21 days. And again, you see very nice sarcomere formation of the cardiomyocytes, okay, in addition to good electrophysiological properties of these systems. Again, this is looking at unpatterned cardiomyocytes. You see kind of a very disorganized cardiac culture. So say if you had a initiated conduction here, what you do dispersion losses that went away. But by patterning, what we do, we line up the cardiomyocytes on top of the patterns or in the cantilevers, so I can look at conduction velocity over a short path length, long path length, and have the same conduction velocity. And that's illustrated here, where you see the long path length, the short path length, same conduction velocity for both. We've done well over 50 drugs in terms of safety in these model systems. We're looking at norepinephrine. We can speed up the heart. We can dose response curve on that. Sotalol, we can actually look at the QT interval changing with time. And we can also measure QT interval numerous different ways. We can also culture out hepatocytes in these systems. This is a 2D for 28 days and 3D for 28 days, getting good CYP activity for 28 days. And also induced activity. This is published in Biotechnology Progress. I wanted to show this because we can then link up very easily our cardiac and liver system. AstraZeneca had approached us. They had a compound that flew through preclinical as soon as they put it into an animal, so had a large QT effect. So they said, well, okay, let's do a validation with terfenadine and fexofenadine. Terfenadine was an antihistamine that was pulled off the market because it caused a long QT effect. However, it's metabolite, fexofenadine, now marketed as Allegra, does not. You can see here a heart-only system, you see a large QT effect. We have the heart-liver system, you see it shifted far to the right. Conduction velocity is not affected if the liver is present. With heart-only, you see an IC50 at 33 micromolar. And again, we're not killing the cells. We are not looking at death markers. The variability is not changing. Now, I wanted to show this also because we can then grid up the system, be able to do modeling, to be able to predict the absorption, be able to predict the flow rates, the steer stresses in the system. We can model absorption by actually measuring the compound in the system before we start experimentation. We can also then look at low absorption, high absorption. Terfenadine absorbs highly, fexofenadine does not. We can also look at mixing in the system. We can look at metabolism. So we can then get area under the curve. To be able to do PK/PD modeling in these systems, you really need to get the exposure profile, which 95% of these systems are as dosed, but we can get the area under the curve with these model systems, which allows us to create a PK/PD model where we can actually account for absorption, be able to pharmacokinetics of that, metabolism, pharmacokinetics of that. And again, because we have the clinically relevant functional readouts, we can get the pharmacodynamics, okay, of the system But now since terfenadine acts on the hERG channel from inside the cell, we can actually add in a different compartmentalization aspect of it, and then be able to get the true field potential duration in the model system. We use that data to predict guinea pig, dog, and non-human primate data. This was published in Nature Scientific Reports with AstraZeneca. Now, with their compound, we did actually were able to predict the QT interval with the heart liver, but we also saw with the heart, we saw an effect which we didn't understand. But what we actually determined was that the cardiac cells themselves were causing the enamine hydrolysis of the compound, and we're able to do that in this model systems where we can actually then look at not only metabolism from the liver, but metabolism for the additional tissues as well. We can also add in GI tract, which I won't show how, which also has its own CYP activity as well. And again looking at drug combinations, we can look at risperidone and see how that affects the QT interval, okay, in the model system at 2 hours. We can see how that affects it, okay, at 12 hours, and at 24 hours, looking at the field potential duration or the QT interval of these model systems. And then again, use that to be able to look at drug-drug interactions in this model system, again, looking at 2 hours, again, looking at the effect of multiple drug-drug interactions in causing the elongation of the QT interval. Now, a new paper that has been under review right now, we've been able to show, you ever wonder why somebody who looks perfectly healthy goes out and starts shoveling snow, has a heart attack? Well, one of the things that you might consider is this idea at rest, okay, a drug has a certain profile in terms of QT prolongation. However, during exercise, that QT prolongation can actually then increase. And you can see here, based upon exercise, okay, the drug, apparent drug interaction, okay, gets higher. Looking at the maximum heart rate, you can basically see here that your administered dose now becomes unsafe, okay, at lower concentrations. Okay, this is something that we are offering now as a surface, again, through Hesperos, okay, the model system, looking to see the effect of exercise on drugs in terms of heart potential toxicity. Now, Roche had asked us whether we could take that system, look at both efficacy and off-target tox in the same system. So we looked at the liver, the heart for both electrical activity and also force, looking at a non-multi-drug resistant cancer and a multi-drug resistant cancer. And you see here, tamoxifen by itself is not a good chemotherapeutic. With liver, you do see with the non-multi-drug resistant cancer, you see decrease in proliferation. Same thing with the metabolite. For the multi-drug resistant cancer, you see from tamoxifen, with the liver present, for the metabolite, nothing, but you add a second drug, verapamil, you do see that decrease in proliferation. No additional loss of proliferation due to verapamil, showing it is not acting as a chemotherapeutic. It's simply just blocking the PGP channel, which is pumping out the chemotherapeutic as fast as it goes in. Now, this is very interesting in terms of looking at tamoxifen. You see conduction velocity go down, but recover. Force go down, but recover. Beat frequency go down by a little bit, recover. But for the drug-drug combination, you see conduction velocity go down, but not recover. You see cardiac force go down, but not recover. But now beat frequency, you see go down and recover. Now, if you were just looking at beat frequency, say, well, maybe this is toxicity I can live with, okay? But then as soon as you put into an animal, the heart would stop working, and you would basically have to start over. And again, this is all done without cell death. This is published in Science Translational Medicine with Roche. Now, recently, their drug was being held up for an IND review because they're asked to do a 9-month beagle study. So what we did is we did a 4-organ system with their 3-month beagle study, and then actually was on review. And then once that was actually reviewed, it lifted the hold on that, and then it was approved, the FDA recently, for motion sickness for Vanda Pharmaceuticals. I won't go through all the data, just showing you here. We can get the liver, cardiac cantilevers, cardiac patterning. We also have a long-term potentiation model, which we do with a human cortical neurons. We can actually monitor that. We also monitor the effect on kidney in this multi-organ system, and we're able to determine the data that made the FDA comfortable to lift the hold on their IND review. Now, part of that work, doxorubicin was one of our controls. You can see here with the cardiomyocytes, after 9 days in culture, everything looks good in terms of morphology. About day 11, you start seeing a problem. And by day 14, okay, you do see start the disintegration of those patterns. But if you're looking at the conduction velocity, you see the effect very early on, okay, when the morphology still looks good. And again, I would point out that the cells are all still beating here. So if you're looking at just simply a organoid looking at cardiac beating, this would not give you the true picture of what it would eventually do, okay, in a in vivo setting. We also can do ischemia in these model systems. We created ischemia boxes in the models where we can look at both the force and also the conduction velocity in the system. This is just data that we have that we published with Zealand Pharmaceuticals, again, showing that for the control, we get good properties. We're able to induce ischemia, but we're going to show that with their compound, we're able to reverse ischemia, and this is now an approved therapeutic. Lastly, we can also then take this system and be able to put in immune cells. This is just monocytes you see circulating in the model system. And what we did in this model system, looking at the cardiac, skeletal muscle, neuronal, and liver, we were able to then treat it with a therapeutic that would then damage selectively amiodarone, the cardiac cells, and we saw the M2 or restorative phenotype, and we added in interferon gamma and LPS to be able to simulate a viral and bacterial infection, and we basically saw that all the organ systems were damaged, okay, and we got the right cytokine profiles for that. This is published in Advanced Science with Roche. So I wanted to go through a lot of information. As you can see, though, most of it's all published. I'm happy to share that with you, and any additional information, please contact us at Hesperos. You see the link here, and happy to answer any questions.
Thanks so much, James. We don't have time for any questions right now, but if anyone has any questions for Hesperos or James, please go ahead and put them in the Q&A box. But thank you so much for your presentation, and we'll go ahead and bring up the next presenter, Vincenzo from STEMCELL.
Okay, so today I want to talk about what STEMCELL Technologies can do and offer in terms of standardized products and reagents to help this workflow, which the speakers have been talking about, starting with iPS cells and then generating different cell types. And today I'll be talking about cardiomyocytes and then doing all these N-type assays like drug screening and toxicology. So what we do here is have a set of products that start with media that can create different types of cells, which I'll talk about, ventricular atrial cardiomyocytes. We can expand those cardiomyocytes. We have products to replate them and dissociate them, and also to freeze them. This is a typical workflow that we do. It's about a 15-day workflow starting with PSCs, and then using our media, you can direct differentiation through an HPSC modulator, and then differentiating them through mesoderm, cardiac mesoderm, progenitors, and then eventually to cardiomyocytes. So we have products that can make ventricular cardiomyocytes that have this type of morphology and beating characteristics, and atrial cardiomyocytes, which you can notice beat quicker. So why is this important? As we know, the heart has two chambers, functional chambers. So we have atrial and ventricular. Each of them have a unique electrophysiology. The atrial exponential is shorter, while the ventricular exponential is longer. So in black are ventricular cardiomyocytes made with our kit. We can see that they beat regularly. They have a prolonged FPD, as well as compared to the atrial cardiomyocytes. We can see that they beat faster and they have a shorter FPD, which is why they beat quicker. If we look at bulk RNA-seq, so looking at atrial cardiomyocytes and ventricular cardiomyocytes at two different time points, day 15 and day 30, we can see that atrial transcripts, which would be upregulated like KCNG3, J3 PIDX2, KCNA5, they're all upregulated in day 15 and day 30, while in ventricular cardiomyocytes, they're downregulated. And then For ventricular transcripts, for example, MYL2, HEY2, IRX5, these are all upregulated at day 15 and day 30, while in atrial cardiomyocytes, they're downregulated. So how can we use ventricular cardiomyocytes made from HPSC for cardiac toxicology testing? So as discussed in previous presentations today, the lengthening of the QT interval is important because it can lead to Torsades, and there's a host of ion channels which regulate this cardiac action potential, and hERG is the target for a lot of these drugs. So you can classify based on risk tolerance drugs, and I have three here, for example, nifedipine, which is a low risk, which blocks the alpha calcium channel, STEMCELL, which is a medium to high risk, which was withdrawn, was an antihistamine, which blocks hERG, and quinidine, which is a multi-channel blocker, and it's a high risk for Torsades. So using our ventricular cardiomyocytes and using an MEA platform, we can see here with nifedipine, looking at FPD relative to DMSO on the y-axis, as we increase the concentration of nifedipine, we don't see too much change until this higher dose, but it doesn't reach this threshold for FPD lengthening. For STEMCELL, we can see that as we increase the dose, we reach this threshold, and then it goes above it. And with this white bar, it refers to the presence of arrhythmias or quiescence, so that the culture stop beating. And quinidine, we can see that as the dose increases, we reach this threshold earlier, and that we see much more arrhythmias and quiescence happening. So the next thing that we wanted to test is that can we use ventricular cardiomyocytes made from these human pluripotent stem cells to do a drug study. And so what we did is we looked at a classic blocker, open channel blocker called hERG, blocks hERG E4031, and it's a poor blocker. And what we see here is that after 10 minutes of dosing, that we get a prolongation of this field potential duration shown in red. So the next thing that we want to do is look at an activator of hERG, RG3, which was published a few years ago. And what this does is that it binds to a different part of the hERG ion channel. So E4031 blocks the pore, and RG3 attaches to the voltage sensing domain. So what we wanted to know is that with this blocking effect through E4031 and this activating effect, could you rescue the E4031? And so what we did is that we first had a baseline recording in black. We perfused E4031 here, and we get a lengthening in gray. And then with RG3, we see that we get a partial rescue return to the control level with RG3. So what we want to do is look at beat period and see how the beat changes over time. And so this is the control. And then with the beat Poincaré plot looking at E4031, we see this irregularity in the beat period, which is expected over time. And then with RG3, we see a rescue or partial rescue return of the normal beat period back to baseline. So this shows that using these cell models that you can look at drug interactions. The next thing I want to talk about is a product that we have on the market for the last couple of years, where we can expand ventricular cardiomyocytes in 2D. And what happens is that we differentiate ventricular cardiomyocytes. So on day 11, we start proliferating the cells. And what we see here is the purity of the CTNT increases over time. This is Ki67. This is increasing, and these are background cells, which are not changing. So we can see that the Ki67 increases over the first couple of weeks and then comes down. So what happens over time is that we have a low seeding density. We see an expansion from day 1, day 2, day 5, and day 7. So these cardiomyocytes are proliferating. The greatest fold expansion occurs over the first 2 weeks. We see a cumulative fold expansion of about 80-fold, and that over these 4 lines tested, we see that a progression in purity from 80% to almost 100%. What we notice that when we expand these cardiomyocytes is that all 4 lines, which are genetically different, they all kind of resolve to this slow beat rate, which is about 10 beats per minute. And they all started around 30 to 60 beats per minute. So this is an example here. So what we noticed is that looking at each passage over time, so that's in yellow, so as you expand, and then in gray is just keeping the cells in culture. We don't see much variation in the beats per minute. However, when you expand the cardiomyocytes, we see that there's a regular drop in beat rate to where it kind of plateaus here at the 4th and 5th passage, so that we get this kind of resting at 10 beats per minute. So what we looked at using bulk RNA-seq is looking at the expression of pacemaker genes, so HCN channels, HCN1, HCN4, and a T-type calcium channel, and that we can see that on day 11, this is the start time, and then if you just keep the cells in culture without proliferating them, we see that they kind of stay elevated over time. However, as you expand and proliferate the cells, we see a reduction in these pacemaker genes, which correlates to the reduction in beat period. Another interesting thing that we found was that maturation markers also improved. So here's start day 11. If we just culture the cells, they kind of stay down, these maturation genes. However, as we proliferate the cells, we see that these genes are increasing. And then we want to look at a comparison to human ventricle. So the human ventricle is here, the adult human ventricle. And a neat finding that we saw was that in control that the hERG channel and the L-type calcium channel stay down regulated, and KCNQ1 increases as you just culture the cardiomyocytes over time. However, when you expand the cells, we see that the hERG and the L-type calcium channel both increase, while KCNQ1 goes down, which is similar to what you see in the adult ventricle. So what we think is happening is as you proliferate and expand these ventricular cardiomyocytes, you're adopting a more mature phenotype. So in summary, what I've showed you today is that we have products that generate specific atrial and ventricular cardiomyocytes. You can use those cells to do predictive cardiac toxicology. You can do drug discovery interactions. I showed an example using the hERG ion channel, using a blocker and activator. We can expand these ventricular cardiomyocytes, which advances maturation, and we think that these standardized protocols ranges can help identify cardiotoxic drugs and help in drug discovery. So thank you. That's the end of my presentation.
Thanks so much.
So I hope everything can see my slides.
Yes, I can see them.
Okay, first of all, thank you very much for joining everyone, and also having me here as the last presenter for today. So I'm either in between your lunch break or your dinner, and I hope you still have enough blood sugar to follow. I'm from InnoVitro. We are a contract research organization, and we are decoding cardiac drug liabilities using human cell-based assay systems. And the way we're using this is, I think, fitting quite nicely into the global momentum that we have at the moment towards a more human-relevant drug development. And for those, I've picked like some of the key players that are around at the moment. Of course, there are many more, which is the EMA, the FDA, and of course also the OECD for like in general streamlining for having all the same goal of reducing the animal testing by focusing more on the human relevant models and thereby also implementing those in the regulatory guidelines for the FDA and for the EMA, for example. And we already heard from Elisa, for example, some programs where we can all put our great technologies in iStent, which is covered by the FDA, which is like focusing on the NAMS, but also we have general novel methodology qualification programs like the EMA. And besides of those programs, there are also some international scientific consortia that are there, which are focusing on special sub-questions. For example, the HESI chronic cardiotox study, focusing on the idea of, okay, can we use NAMS for addressing chronic cardiotoxicity? Or the Botanical Safety Consortium, focusing on botanicals. Do we need guidelines for them because they're being used so much? Are they actually going towards toxicity? And also, when all these disease modelings are coming up these days, can we also focus on atrial fibrillation? What is the NAMS in there at the moment? What is the current state of the art? And the reason why I put these scientific consortia on this slide is because I want to show you where we are participating with our technologies in the cardiac field and how we are implementing our assay systems to show people how we can use them. And before I go into depth about some case studies, I would like to give you an overview about what we are focusing on. So basically, we are focusing on three major pillars: the electrophysiology side. When you think of the cardiac contraction cycle, we have the beginning with the electrophysiology, but then we come in also with the contractility, where we basically then get the full picture of the cardiac contraction cycle, have the functional performance on the one hand, and if you like them, we can also combine that with the morphology. So here we have the structural assay that we then check towards the functional to dissect if we have functional or structural or even both on a toxicity level. Technology-wise, we use for the electrophysiology the cardioexcite system, which is an MEA-like system on a 96 well level, also recording extracellular field potential duration, so focusing a little bit on QT interval. And here I will show you a case study where I thought, for those electrophysiologists out there, pro-rhythmic risk is the thing. So QT interval changes, as we have heard of from some of the great presentations before already. But here I wanted to actually make the dive into the frame of the atrial fibrillation, because you can see how we can actually, with the technology, shorten the QT and then with the rescue effect, prolong the prolongation again. Then I will follow up with the contractility, so focusing on force analysis, where we have a nice physiological environment with membranes on a 96 well level. And here I will focus a little bit more on the contractility on a chronic level. So we can do acute, of course, but we can also look on the contractile liabilities that will pop up later in stages, like a couple of days later, to see different effect kinetics, to see that time has also a strong effect on the toxicity. And last but not least, I will combine those with the idea of the morphology, as I said before, with the cardioexcite system, again, choosing the impedance-based cell monitoring and dissecting the functional liabilities from the cellular damage. All our technologies we're using are completely real-time monitoring, so we're completely non-invasive. There are no dyes involved, and we go high throughput with 96 wells, so we can do a lot of screening, especially when it's more like early in, when you do a lot of higher drug screenings, and we are pretty physiological, especially with the contractility assay that I will show you a little bit more detail about, but also with the idea of going study dependently from 2D monolayer up to organoid level, because it basically creates, when the project comes in and we have a discussion with a client about how to design it, there are a few components that are obviously taking part into the decision-making of, are we staying 2D or are we going organoid? And this is, of course, once the translatability, but also the throughput and the cost, because the higher you go with the organoids, the more cells you're using, and that will, of course, also affect the costs. And that's also a key player when you frame your design. And in the end, you'll find a great sweet spot for doing either a safety request or doing toxicology study with us, or the efficacy testing. We also do the cell line characterizations, because there are many people out there, many pharma companies, biotech companies, having their own disease model or having their own normal human iPSC-derived cardiac cell line, and they want to address that and see that they're functioning right, and then they come to us. And of course, we have the disease models that are coming and that we address. So this is something that I will pick up now to go straight into the first case study, where I already told you I would like to show you the atrial fibrillation disease model and how we're using that, basically how we're testing for the QT within this model. Here we use atrial human iPSC-derived cardiomyocytes from Axol Biosciences, so commercial chamber-specific cells that we plate on our specific NSP plates for 9 days. And the well design is that per each well, we don't have, like with the MEA system, several electrodes. You have one big electrode, but you also have the reference electrode, of course, and the stimulation electrode. And with the stimulation electrode, you can perform the tachypacing that we need to do. So in order to receive the electrical remodeling that you would usually see with patients with AFib, we perform a tachypacing procedure for 24 hours on these cells with 2.5 hertz and see in correction to that the EFP duration shortening, which is, if that would be the first pacing and the second pacing, a shortening of this EFP duration here. And this is then something we try to rescue with a class 3 antiarrhythmic drug, tofetilide, and the data I'll show you now is the first one, how we saw the reorganization. So you have to imagine the 100% here is when we started the tachypacing, and you see that within a few hours we have a nice decrease. So basically a shortening of the EFP duration, an analog for the QT prolongation, and see that we have a very stable shortened duration around 60% after, let's say, 5 to 10 hours, and it stays stable over time. And then we kept on doing the tachypacing and added the compound tofetilide in different doses. And you can see that with the highest dose of 10 nanomolar, we reach a very nice increase, rescue effect, and stretch basically the shortening, prolong it again, and go back to almost 100% again. So having this nice rescue effect. So for those of you working with AFib model, maybe you want to have a human-based, cell-based assay system work with us, or the ones who are actually thinking of the QT prolongation, arrhythmia, etc. That's also something we cover nicely here. So the next step would be going towards contractility, where we will look closely at the delayed oncology drug-induced liabilities using commercial iPSC-derived cardiomyocytes. But first of all, I'd like to go a little bit deeper into the technology, because here we have still, again, a 96-well system, but we have the membranes instead of the plastic surfaces. So we have only 4 microns in thickness, by the way, 33 kilopascal in stiffness to reflect the human heart conditions. And then we plate the cells on top. They can be, like I said, 2D, they can be organoid, both works. And when the cells are on top, you see basically the plate here from below. And each well is hanging through a little bit because of the weight of the medium. And the electrodes down here are actually part of the device. And when the contractions of the cells are happening, they lift the membranes upwards a little bit. And this change in distance is then measured via capacitive distance sensing. And these are all the contraction parameters we can measure. Of course, proper contraction force, beat rate, irregularities, arrhythmia. We can do beat duration, contraction, relaxation slopes, and area under the curves. And for those of you who are very known in the electrophysiology field, all the things that you can see there are being basically translated into the contractile motion as well. So for example, if you would have an early after depolarization, what you would get on a contractility level is an early after contraction. If you have QT prolongation, you would see a beat duration prolongation. All these things are being translated, and therefore you get a nice overview of what's happening in electrophysiology level is being translated into the physiological motion. And this all on a very high throughput. Okay, just jumping straight into the chronic cardiac liabilities performance that we have done, funded by the CAAT, so the Center for Alternatives to Animal Testing, a while ago. We focused on oncology drugs, so anthracyclines, tyrosine kinase inhibitors, mTOR inhibitors, and tested them over 5 days and tested in 4 different doses. So we have the different doses ranging from 10 nanomolar up to 10 micromolar. We have 1 day, 3 day, and 5 day here depicted for the heat map, and the parameters for speed rate and beat duration. The seizing effects are highlighted in red, and in turquoise we see no effect and increasing effects in purple. And what stands out here very nicely is if you look at the anthracyclines, we have, dependently, of course, on a dose and a time-dependent increase in toxicity happening for all of these parameters, showing you that chronic analysis is indeed very important to show the in-depth toxicity that can happen. If you look at, for example, cardio-safe compounds like allotinib, you can see that even at longer time exposures and at higher concentrations, we barely see an effect on the contraction force or the duration or the beat rate. But of course, it's not only a heat map that we can do, we can also go into very detailed analysis and check actually for the different effect kinetics that are happening over time for the different compounds. So for example, for venovelbine, we see a very clear dose-dependent effect where we have an immediate reaction upon administration of the compound, but then a very stable signal over time of 5 days roughly in this case. If you then look at doxorubicin, we see actually both. We see that the cells are directly affected by the doxorubicin, but also that time plays a role, and we see a progression profile over time. And lastly, when we look at the BMS compound, we see that we have very linear kinetics of the different doses, showing you from going basically decreasing until ceasing, but very linear in their whole shape. So very different effect kinetics that we can basically spread out and look at in more detail. Okay, coming to the last case study, which is dissecting the functional liability that we have from the cellular damage that might happen if we have a look at it. Open a door basically to the idea that it's not only functional. So if we are having the action potential and the contractile motion as the parameters we look at on a functional level, what we then do is compare that with the base impedance that we receive via cell attachment morphology and viability. So it works similar to the well that I've shown you before with the atrial fibrillation model with the electrode. So there's a current applied, and when the cells are plated on top, they basically impede the current, and thereby the cell signal here is rising to a plateau when the cells have reached a monolayer or a status quo where they're healthy and fine. And when you then add a compound that would have an effect on the cell, if it would be toxic, the cells would be dying and swimming off, the signal would decrease, or if the cells would be swelling or proliferating, the signal would increase, and therefore giving you a nice idea, an indication of what might happen here. So the first thing I'd like to show you is comparing the contractile and the morphological data of doxorubicin. It's such a nice tool compound, especially when it comes to toxicity. So what you can see here on the left-hand side is the spider plot of our contraction analysis. It's just a nice way of putting a lot of parameters into one figure. So it's the contraction force, the beat rate, the contraction and relaxation slope, and the beat duration. And you can see that the highest concentrations, this is time point 3 days, by the way, are already knocked out by then. You don't really see it anymore, but the lower concentrations show you that basically all the parameters are affected in a concentration-dependent manner. And if you then look at the at doxorubicin on the base impedance parameter, just imagine that we have a plateau here where we administered the drug, then you can see that we have a concentration-dependent decrease in base impedance for quite a bit, down to 40%, showing you clear toxicity in a dose-dependent manner. If you then look at another compound like sunitinib, for example, we have the similar outcome here with the spider plot for the contraction, for the contractile properties, where you can see that we have a very strong effect of sunitinib on the beat duration and the beat rate, like a decrease in beat rate and an increase in beat duration, basically the higher the doses will get. And if you then look at the base impedance parameter, you see actually the exact opposite effect. You see that the lowest concentration is similar to the functional effect, has basically no strong effect on everything, and similar here to the base impedance, nothing is really happening. But then we have a dose-dependent increase, an immediate reaction to the base impedance. So not showing toxicity in a way, but maybe opening the door to the idea that these cells are either hypertrophic or maybe having proliferative capacity, because the tyrosine kinase inhibitor does affect receptors that would be involved in proliferation. So, I'd open a door to the possibility that some things are changing, which have to be looked in further with further studies. And with this, I'd like to summarize what we have done here. So how we can basically use us. If you want to get a nice mechanistic resolution of human cardiac liabilities, you can come to us for the QT prolongation shortening or the atrial fibrillation to identify early proarrhythmic risk. You can then combine us for, if you do that at home, for example, we have a lot of customers that actually do their electrophysiology studies in-house, but then do the contractile compartment as in a partnership with us. So we can assess the acute as well as the delayed functional cardiotoxicity on the contractile level, and also then combine that with our structural toxicity test to give you a window of if anything is happening on that level and if you need to look at it further. And with this, I'd like to thank you very much for your attention, and I'm open to questions.
Thanks so much, Bettina. So if I could have all of the speakers come back on camera, but stay muted for the Q&A, we can go ahead and get started. There's already a few questions to go through. So the first one is for Lizzie. Can you describe how the system can be used to conduct shear-based experiments using external pumps? Are you able to simulate in vivo, i.e. coronary shear conditions?
Yeah, and thank you for that question. So if you wanted to add flow continuously and actually monitor the shear that would be on the blood vessels in culture, you can use the Identix 3 and 9 platforms because their media channel inlets and outlets are able to be connected with luers, which then can connect to tubing and off plate syringe pumps or peristaltic pumps. And so then in that way, you can now regulate the flow rates and shear stresses on the cells. And typically that would be in the range of like 1 to 10 dynes per centimeter squared, which is sort of like the levels of in vivo wall shear stresses in coronary arteries. And yeah, happy to continue the conversation if there's any more specific model requirements needed.
Oh, thanks, Lizzie. This next one is for James Hickman. You showed some great pictures of cardiomyocytes seated in perfectly straight, thin lines onto some electrodes. How were you able to seed those so precisely?
Well, we do surface patterning. So what we do put down a mon layer, we shine laser on it, and then we come back in with a second compound, and then the cells will grow on the positive surface and be repelled by the negative surface. So we can pattern any kind of geometry. We can look at reentry, all kinds of various aspects using the surface chemistry, which is kind of my roots. That was my original training, was as a surface chemist. So
cool, thank you. I think this one's for everyone. I was wondering how important it is the use of serum-free medium, and what are the pros and cons of different cell sources, cell lines, primary cell, iPSC derived. Thank you. So if anyone feels compelled to answer this one.
Right. So we've been working in serum-free media for a very long time. What does it? Gets rid of a variable out of your system, right? Because with calf serum, it's different all the time. I mean, back in the day, you had your favorite bottle of calf serum you used because the cell experiments work with that one, with not another one. But using a serum-free media makes it very defined. But also, if you're really trying to do PK/PD and you're trying to looking at analysis of a compound in terms of concentration over time, serum really interferes with that quite a bit. By having a serum-free media, it's much easier to measure the compound concentrations to do PK/PD.
Thanks. Anyone else?
I would agree. We use our main goal is always to develop defined medias that are away from serum for the points that James discussed. So really in the discovery process, those undefined variables can affect a number of things. So defined media for sure is the way to go, and also for keeping the quality of the cells, reducing variables.
One other point I did mention was that fetal calf serum basically has all the factors in it for it to be a calf. Okay. And one of the things that people found is they used primaries and they de-differentiated. Well, that's because you put it into calf serum, it's basically telling those cells you need to go back to an earlier phase of differentiation. As soon as we remove the serum out of it, we can maintain primary livers out for 28 days, and we published that. And the same thing with the cardiomyocytes. We could get them to fully mature over time in a serum-free medium, which is not possible with the serum-containing mediums.
Okay. Another general question for all speakers: how should our field think about statistical sample size related to biological versus technical replicates when using iPSC-derived models? For example, what do you consider as the sample size if you take 5 different iPSC lines, then drive each cell line into cardiomyocytes across 3 different batches per line, then make 24 individual tissues per batch? Curious to hear how different groups consider this.
Well, you must have a lot of money to do that. Well, what we find is it's how strong is the effect you're seeing, right? Normally we try and do 3 different experiments, 2 replicates per experiment. That normally will give you statistical significance in the model system if your effect is strong. If you're looking at subtle effects or trying to see small changes and things like that, well, then obviously your sample size gets a lot bigger. If you have a very large effect you're trying to see, you potentially get rid of, you get by with less replicates, less experiments in the model system. It really depends upon, what exactly you're trying to monitor in terms of the sample sizes you need to be able to develop in that system. And again, as I mentioned first, it's a cost driver.
Yeah, I agree with what Jay was just saying. I think it depends a little bit on the response, like you mentioned. And then 2, I think like an academic research publication versus maybe a drug study are going to have some differences in terms of statistical power that you would need to take into consideration. But I think the general rule of thumb would be 3 biological replicates, so like 3 fully separated experiments, and then within each test condition having usually like a minimum of 3 technical replicates. But I think it depends a little bit on the consistency of the data. If you have very variable responses, then you might need to run more technical replicates in order to make sure you actually are observing the right effect. Yeah, that's how I think we were thinking about it too.
Anyone else want to comment on this one?
I guess I'd be curious to hear from the panel, because I think another question kind of in the same vein is around donor variability and how many donors would you be required to run to have sort of a representative sample of whatever population you're looking to study. So I don't know, I get different responses depending upon who I talk to, like maybe in the range of like 5 to 20 donors. Again, it's dependent on the availability of the tissues that you're interested in studying. But any thoughts on the donor availability from the panelists, from the speakers?
I mean, for sure, I mean, what we do when we're developing products in R&D here at STEMCELL, we do a minimum of 4 different donors, so IPS donors. So and that's male and female as well. So the reason to do that, a lot of our products are geared around understanding development and then kind of controlling development to a certain phenotype. Today we talked about cardiomyocytes, but we do the whole, all different type of organ systems. So in general, 4 is the minimum, but we do as many as, for some cases, just because we've had disease lines and other things that have come in, we've gone as high as 10. So, but I think having the different donors, because we do see that they can respond differently, so they don't totally align. So, but which is why doing the sequencing and those other types of checks along the way is important. So we always at minimum try to use four different donors.
Yeah, it depends upon the question. When we did the study for Vanda, we did a low metabolizing liver, high metabolizing liver, and a medium metabolizing liver. So we did 3 different donors, but we pre-screened them. We screened about 20 to be able to get in the right ranges of what we wanted to try to do. But trying to run, experiments on all those different donors, a full experimental panel would just be hellishly expensive. So it's always a trade-off with cost versus benefit in these model systems. Again, complexity versus not in these model systems. It cost is a driver for all of it. So you can always design the experiment you'd love to do, but as soon as you draw that up for somebody, they say, "Whoa, we weren't expecting it to be that expensive." And so it's a trade-off in terms of what you're trying to figure out. But again, what we do a lot of times is start off with the least complex system and then building complexity as necessary. Where and so you can probably get by with smaller number of donor lines. And then there's the whole idea of pooled lines. In liver, I know that does people do that. I haven't seen it so much in induced pluripotent stem cell derived systems, but I guess you could do it with different donors and be able to look at a pooled system. That has certainly cut down your costs quite a bit. I'm not sure how you would differentiate that in terms of being able to characterize it, but I guess that's also another method that people have used in terms of looking at different donors.
Oh, anyone else?
So, yeah, what I would just like to point out from our perspective is that we usually are more like on the receiver end, and we work together with the commercial cell type providers that are usually don't provide that many donors when it comes to like the healthy cardiomyocyte. But then what we do is to assure to our clients that this is working very nicely, is do lot-to-lot studies to show the different the stability of the donor itself when it comes to screening, and also I think as soon as it goes to disease modeling and then having the different donors, it's like it's been mentioned before, definitely a cost thing that is a big decision maker here.
Yeah, maybe just small addition here. So if you have a genetic disease, it's always advisable to use some isogenic controls. So you have like mutation specific effects that you can measure. Then you can reduce the amount of donors that you're checking. But if you have like sporadic disease where you don't know exactly the cause, then of course then the at least 4 to 5 different genetic backgrounds, I think, would be reasonable to take along.
Thank you. Okay, another general question. How well are these platforms validated for use with modalities other than small molecules, e.g. ASOs, protein degraders, particularly around absorption, uptake, delayed onset of toxicity, etc.?
Well, we work with biologics, we work with ASOs. The study we just did, Vanda Pharmaceuticals, that was published was an ASO, and that's now in clinical trial for treatment. We do antibodies. We published that with Dianthus, and that's actually now is in a phase 3. So we can use peptides as well as small molecules. So we can look at absorption, we can monitor all that, and do PK/PD for it as well.
Yeah, I would say generally the platforms, at least from AIM Biotech, are modality agnostic. So whether it's a small molecule or any type of biologic or even cell-based therapies, it would all be compatible. And it's just maybe a matter of what's the end target there that you're looking at. But the delivery, at least in our system, is through the media channel, and then it goes into the hydrogel compartment where the tissue actually is. And so that's where you could then monitor the responses. Using imaging, or you can collect effluent, you can collect the RNA at endpoint and do different things to see how whatever compound it is that you've added is affecting the ADME properties of the cardiac microtissues.
Yeah, in our platform we use 3D tissues, so we have tested small molecules, gene therapies like AAVs. ASOs is still, and DNA transfections we're still in testing, because of course you have the 3D structures to make sure that you have a efficient transfection and transductions. Some people do first transfections in 2D and then they embed them in 3D. But it also depends on the modification of ASOs. So some are more efficient to penetrate the 3D structures, some less. So we are still looking into that, but it's a very important one. Yeah.
I think this next one is fairly similar. Any challenges or concerns of recombinant protein and antibody penetration to organoids? Similarly, any shuttles would be required for siRNA approach, especially to cardiomyocyte constructed 3D systems?
Yeah, I could jump in. I think as you start adding in different elements to the model for cardiac tissue, so for example, like I was talking about adding in things like vascularization, there's also extracellular matrix surrounding the cardiomyocytes if they're the target cell. And so in particular with siRNA, to make sure that it's stable during the circulation through the media channels and then into the vasculature, it can actually get out of the vasculature and move through the ECM to get to the target cell. I think a shuttle would likely be needed. And it sort of is helpful in representing kind of that delivery process in these more advanced microfluidic 3D systems. But the answer is yes, that you can do that type of study, and you would most likely need some sort of like lipid nanoparticle or other shuttle proteins to help with that delivery process.
Yeah, again, that's one of the beauty of the multi-organ systems we have is that you can look and see the recirculating system. You can calculate the PK/PD for that. But we just published the first MPS digital twin model for malaria, where what we can do is it's a cost issue with a complexity. So what you can do is sometimes you can measure certain properties like vascular transport, separately, and put it into an in silico model to be able to predict what's going on. So that's where the digital twins become very, very important, okay, for these modeling systems.
Any other comments for this one? This is our last question currently, so if the audience has any more questions, feel free to put them in the Q&A.
Well, I just put a little shameless plug for the MPS meeting in DC coming up. Please, if you aren't aware of it, if you need information, you can email me or the MPS Society. But we're having a meeting in DC the last week of May, where everybody on this call, I think, is represented there. As sponsors, we have a large program in Brussels. We have 1,500 people, 125 exhibitors. So if you're more interested in these model systems, you want to talk to people directly, see demonstrations, please think about coming to Washington, D.C. for the IMPSS Summit.
All right. Doesn't look like we have any more questions, so I'd like to thank all of our speakers for being here and all their great presentations and answering the questions. And yeah, thanks to our audience for being here, and I hope everyone has a great rest of their day. Thank you so much.
All right, take
Thank
care,
you for
everybody.
the
Bye-bye.
invitation.
Good to see you all.
Thank you.
Bye.
Bye-bye.